What we've built at the University of Pennsylvania through the Center for Cellular Immunotherapies is an integrated team to facilitate the bench to bedside transfer of novel cell, gene, and biologic therapies. We have an array of research laboratories that are engaged in all aspects of cancer immunotherapy research, from the discovery of new targets to the design of new chimeric antigen receptors and switches, to the delivery of genes, the ability to activate cells and maintain their function, and to translate those from in vitro models to in vivo models in humanized mouse models, and then to scale that from millions of cells to billions of cells to enable the manufacturing. We have a manufacturing team that can also do the testing of the cellular products, a correlative studies team that is able to take samples from patients after infusion and learn everything from patients who respond and from patients who don't respond, so that we can re-engineer the therapies to make them better. Supporting all of that, we have a clinical operations team, quality assurance, regulatory support. So in effect, we have built a mini biotech within the University of Pennsylvania to develop these new therapies. We have developed a platform for T-cell engineering, and with that, the clinical and operational infrastructure to facilitate first-in-human clinical trials. Companies with new technologies, because they know of our expertise in clinical trials, have come to us with new technologies over the years, beginning with the very first lentiviral vector, the first use of gene editing with the zinc finger nuclease. Those companies, Phacilitate and Sangamo, came to us and said, we have this technology, but we need it validated. We need the preclinical work conducted, we need it scaled up. We were able to do that, to conduct the clinical trials, to do the analysis on the patient samples, and to show that not only is this technology feasible to integrate into clinical trials, but we can do it quickly in an academic center recruiting the patients. So we serve in a way to facilitate their technologies, but we have developed our own platform, as it were, the T-cell engineering platform, and now we have a spin-out from Penn to Immunity Therapeutics that is taking advantage of that. So not only have we worked with industry from small biotechs all the way up to Novartis over the years, we have done transfer to CDMOs, we have done transfer to academic centers, and now we have spinning out of the Center for Cellular Immunotherapies actually 3 biotechs, Immunity Therapeutics, Charisma, and Cabaletta. So we have become very familiar with the procedures that are necessary to go from discovery to translation to commercialization. So we're fortunate at the University of Pennsylvania to have an academic medical center that is co-located with the premier children's hospital in the world, Children's Hospital of Philadelphia, and together we have both pediatric and adult trials in cancer, in HIV, and we've been able to move the technologies into the clinic because we have clinicians who are expert in those areas, whether it's liquid tumors or solid tumors or HIV. And those clinicians are receptive to these new technologies, and they are our partners in advancing cell and gene therapies to the clinic. So I'd have to say that the academic environment, the clinical environment, the integration, the collegiality, the ability to get things into the clinic, to work with industry, all of those are together in our center, and it's really been a privilege to work with so many investigators who share the same vision that we do. So predictions for the next breakthroughs in cell and gene therapy. Now, is this going to be played back in 15 years? We'll see, but What I envision is that we're looking at cells and T cells and other cells as drug delivery vehicles. We can gene modify them to sense particular environments, to produce a particular function, or even to produce a particular drug. So now think of a cell as a continuous drug factory where the patients don't need to keep taking a bispecific antibody, for example. If we can engineer a T cell that lasts for years, months, years to be able to do that, then we have something that is unique in the therapeutic repertoire, and as demonstrated by the approval of CAR T cells in 2 different indications, a very powerful therapeutic that, while it is paradigm shifting, is now being taken up throughout the world.